Back to PowerPoint.
In this thing. Okay, sorry, everybody online that you missed my spiel about Darwin Day. Hopefully you at least saw the slides. So it's my pleasure to introduce Dr. Fathi Karouia, who is coming to us as a NASA scientist. He did his bachelor's and his PhD at Poincaré in France, in the south of France, close to Strasbourg, I think, maybe. And then managed to go to this international space studies for another master's in Strasbourg, which we heard about this morning. Sounds like a pretty fascinating program. And then decided to go into biology after getting a degree in physics and chemistry. So we're going to hear a lot about his interdisciplinary approaches to space biology today, and I look forward to more. Thank you.
Thank you. Can we hear me online? You got the thumbs up, or are we good? All right, so thank you again for the invitation. I greatly appreciate it. And so a big thank to you, Christina, and the team, Jessica also. Too bad that unfortunately she's not feeling well. Maybe she's online, so then hello, virtually. Anyhow, great pleasure to be here. First time in the area. And I was excited about coming, and for different reasons. I mean, one of which is the fact that I'm always keen to kind of interact with faculty, staff, students, and so forth. But more importantly, also to kind of talk a little bit about space in general, but more importantly about some of the things I've been involved with. And in some sense, I mean, I have two goals here. It's to kind of take you on a journey through space in some sense, and then talk also a little bit about some of the research I've been involved with for the past few years, and in some sense try to entertain you, and hopefully maybe maybe at some point you guys want also to be involved with space research, and so I would be more than happy to be able to talk to you about that as well. So maybe that could be you at some point, and so certainly I think we're living under extremely exciting times, I must say. I think we have an opportunity within our own generation to go back to the moon, but also at some point going to Mars. And so I think that you guys are a lucky generation. And I want, as I mentioned, have a multidisciplinary approach to space research. And I will be talking about a few different topics, four of them, actually. The first one would be astrobiology. And so as you can see, astrobiology is kind of more or less studying the origin, the evolution, and then the future of life. And this is an important component within NASA portfolio of what we call the Science Mission Directorate. And so I will be talking a little bit about that. The second part is related to planetary protection. And so planetary protection is twofold in some sense. One is that when you explore the world, you need to do your due diligence to mitigate contaminations, right? But then also there's a reverse problem, which is backward contamination. At some point, we're planning to have samples coming from Mars to Earth, and therefore we need to protect the biosphere. So planetary protection kind of deal with those aspects. And so I will be also addressing some research related to planetary protection that could be of interest to you from an evolution perspective. Subsequently, we'll be transitioning to the realm of space biology. And so in some sense, space biology is trying to kind of better understand how Earth-based life cope with the space environment and trying to better understand basic mechanisms, but also is an applied element related to it. And then we'll be kind of transitioning towards technology in the realm of technology to enable exploration. And so again, the idea there is to take you on a journey, visiting those four different pillars through some of the research I've been involved with. So again, that's maybe you, but maybe me for now. And then I'll be starting, as I say, from an astrobiological point of view, and kind of looking at acclimation versus adaptation and using E. coli as a model organism. Subsequently, we'll be transitioning to Bacillus pumilus. I mean, this is a Bacillus expert right here. This is different type of Bacillus that was isolated from clean rooms, and so we'll be talking a little bit about that. Then transitioning to the ISS microbiome, so I call that the astrobiome, and so a lot of work related there as well, which will be also entertaining and interesting. And the last part is more or less related to technology and, again, with the realm of enabling exploration. So let's start with, again, astrobiology. And so when you look at the realm of space microbiology, and again, here I'm providing a few elements in some publication of interest, When you expose microbes in space, they behave differently. There's no impact on secondary metabolite. They grow faster. They're more resistant to antibiotics, stress factors, some of which have been shown to be more virulent in the case of Salmonella. Other studies have shown me more recently that if you look at altered gravity, so not just microgravity, but Martian gravity, lunar gravity also change the physiology of the microbes. And then some recent studies also have shown that microgravity enhance horizontal gene transfer. And so, which is all that very, very interesting. The sad part is that the response seems to be cell-specific. You would have think, oh, maybe there's like a gram-negative and gram-positive response to the environment. Doesn't seem to be the case. Every time you interrogate an organism, you can have a different story, and so which is very, very frustrating. But also, again, for me, challenges equals opportunity, and so it's also very, very exciting. And I will be talking about one study that actually I conducted at the time when we were at the University of Houston, which is kind of related to adaptation. And I will be showing that adaptation is both phenotypic as well as genomic. So let's get the grad running. And so when you want to do space research, I think you have few opportunities to do so to kind of mimic the environment, right? Obviously, being in space is the best outcomes that you want to do, right? So microgravity, radiation, etc. But it's not always the case, right? And so there are other things that you could do. So, for example, drop tower in the Bremen's day, actually some people kind of use drop towers where you can simulate microgravity for a few seconds. You can, of course, send a spacecraft to space. You can do those parabolic flights and be on the station, etc. So those are things that, again, kind of mimic the environment, and those are good models to kind of do science. But then also on the ground side, that you have some devices that can be used. Sorry, it sounds like the resolution is not very good. There are devices that you can use to kind of simulate microgravity conditions. So we call that rotating wall vessels. And so here I'm showing an example. So imagine you have a cell which is just right here. If it's not rotating, it will be going downward towards gravity field, right? However, if there's a rotation, then in some sense, the cell is kind of staying and maintaining its position. And so more or less, it's kind of mimicking the low shear stress environment of space. So the study I'll be talking about, we kind of use this system as a system to model microgravity condition. So here is the protocol in some sense. And so what we wanted to do is, up to that time, there was been no adaptation studies looking at the impact of microgravity on microbes. And so here in this case, we're using E. coli. All the studies were only done on short-term adaptation, so growing the cells for a few days and etc. Here we wanted to assess and compare difference from short-term to long-term adaptation. And so to do so, we use actually E. coli as the model organism, but we use two different strains. And the beauty of it is that those strains, which are like plus and like minus, you can actually very easily discriminate them on the plate because one will be orange and the other one will be kind of white. And so that would be very easy to kind of use those two strains and then be able to do what we call fitness assays, where you can compare one to the other. Then the idea there would be to expose those cells to 1,000 G. So 1,000 G is 1,000 generation. So the cells will grow for 1,000 generations continuously in those environments. And either you kind of use the traditional flask in the laboratory, or you use the rotating wall vessels, and then you compare the two. So that's more or less the protocol here. So, and then acclimation versus adaptation is a key parameters there, and maybe some of the students might be interested about getting some insights there. But acclimation in some sense is that I'm kind of fine-tuning my system, so fine-tuning my genes to kind of cope with the environment. Whereas adaptation, I'm acquiring novel functions that is embedded into the genome, right? Those are the two differences. And so the big question is that if I'm exposing those cells to long-term simulated microgravity condition, what's going to happen? That's more or less the question there. And if you do so, then if the adaptation of the strain could be only based physiologically speaking, which means I'm more or less acclimation, or is it more on adaptation role, right? And so what you could do is that, so if a strain is more or less acclimating to the environment, then whatever properties the organism is getting, that will be lost after you remove it from the environment. However, if it's embedded into the genome, then that property will remain, right? And that's more or less the question here. So it is a busy chart here, but kind of going through and explain a little bit all the different conditions here. So control is more or less I'm growing the cells in the flask. So that's my control. here is, I'm doing a competition where I'm comparing using the rotating wall vessels, and I'm continuously growing the cells under similar microgravity condition. And then, here you have the normal conditions. And the idea would be to compete the two, right? And so what we call here one cycle is like 10 generations, so roughly a day, right, where you're growing the cells. And so then the question at the end is that when we remove the strain from the selective pressure, in this case, microgravity condition, what happened next, right? And so then we grow the cells back to the flask for several regeneration and see what happens. Stop this problem. Someone seems we're stuck for some reason, so. Pointer, yeah, it works. Sorry, online, we have some issues with the slide, which are not moving. Yeah, I don't think it's synchronized. Oh, okay. I'm not sure. It's not moving. It seems it's synchronized. Can you go down? Yeah, it's this one. So if I do it. So we apologize online. It seems like the slides are stuck, and so bear with us. Okay. Yeah, this one. Maybe we were not using the right version, I guess. There was different files. Yeah. All right.
I'm sorry about that.
Oh, no, don't worry. I hope online you're able to still see the slides. Make sure. All right, so again, I just described the experimental protocol. So again, ground control, competing, and then we remove from the selective pressure, which is microgravity, and then assess the question how the cells are behaving. So here's a chart looking at a few things. One is that, so there's a terminology. So there's two things there, I guess, to pay attention. One is the average value, which is in white. In black is the ratio. And so we're kind of comparing, again, it's a fitness assay. So we're comparing one strain to the other. So one I've been exposed to the microgravity environment, where the other one I've been exposed to just normal condition. So here, first graph is kind of showing that there's no changes between the unadapted strain and with the adapted strains. However, you can see already here, when we compare the unadapted strain to 1,000 generation, there's a roughly threefold differences ratio. So threefold ratios from the fitness, which means that you have 3 times more cells from the adapted cell versus the other one. And what is interesting is that when you remove the selective pressure, so 10E will be 10 generation after you remove it from the selective pressure 20 and 30, you still have that competitive advantage compared to the ground control. More interestingly, I mean, and I guess it's something I didn't mention. So we had two different conditions. One, when we were exposing the cells to microgravity condition, we had an older version of the device, and that device cannot be sterilized. Therefore, we disassemble it, we kind of mix it a little bit with antibiotics, kind of wash it thoroughly, and then reassemble it, and so forth. And where are the other ones? We had a novel version of the system. In that one, we could sterilize it. What you can see here is that when we try to assess the response of the adapted strain to antibiotics resistance, I mean, many other antibiotics that was tested, many of which, mainly on the bottom part right here, show that actually we have a higher resistance to all of those compared to the one that was using the device which has been sterilized. And so then the question is, maybe our own protocol is like adding a little bit of antibiotics into the system, and because of that, the strains are becoming more resistant. And again, that still makes the questions interesting, because then what you could do is that dive a little bit more. And so there was a following study where, again, we were just emphasizing here on the adapted strands and compare it to when we remove it from the selective pressure, right, in this case. So on the left-hand side here, I'm looking at resistance to different type of antibiotics. I think the key one, which is sulfoxaflines right here. So this is control. This is the adapted strains. And then when we remove it from selective pressure, it kind of go back to reference, which is a good thing to do and to see. Similarly, for cellular, we see exactly that. However, when we come back and we remove the selective pressure, that strain is still resistant. So when we dive a little bit more about using cellular and trying to dive a little bit more, and which is more or less a graph here on the right side, the interesting part is that when we remove it from a selective pressure, so we grow the bacteria back into a flask for 10 generation, 50 generation, 80 generation, 110 generation, is still showing resistance towards the antibiotics. And so it seems like that, therefore, that's kind of remain embedded into the whole genome. And so when we try to ask, okay, what are the different putative mechanisms of resistance? And so when we did the sequencing, and so we saw there was a lot of genes which were removed from the system, which was interesting to see. We noted also there's few mutations, many within genes, some of which were involved with antimicrobial resistance. Also, there's a lot of genes which were used to transposons, which were found into the system. And so that also might have impacted the overall outcome. And then there was also a lot of mutation related to promoters. And so it seems, again, the exposure to microgravity condition led to all those different changes. But more importantly, when we remove the selective pressure, that organism still kind of keep its own properties. And so certainly from a big perspective here, I mean, again, we kind of show that the adaptation was both phenotypic as well as genomic. But in the case of the strain which were exposed to 1,000 generation under symmetric microgravity condition, and most likely the exposure to the antibiotic to kind of wash the system might lead to the fact that that became more resistant to an organism in general. And therefore now kind of raise the question, you know, let's say that an astronaut is taking some antibiotics on some fashion because he's sick in space. How does that impact the microbiome and so forth? And here you can see that, well, those organisms kind of become more resistant. Therefore, that could be a problem. And so that's the first pillar of the conversation. And so again, that was done at the time where we both were at the University of Houston. So and again, very, very thankful for the opportunity to kind of work on that project. So now we're going to shift gears and kind of dive into planetary protection in some sense. And so planetary protection, again, it's kind of, you need to minimize contamination when you build a rover, for example. You build a rover is going to go to Mars. You need to do your due diligence to mitigate that. And so what you do, you build a rover in a clean room. You bombard a rover with UV, hydrogen peroxide. You clean as much as you can and so forth. But as you can imagine, that creates a selective pressure. And it's counterintuitive because you do your due diligence and as much as you can to have as clean environment as possible. But actually, you are selecting for troublemakers. You are selecting for organisms that will survive the trip to go to Mars. And I'm going to tell you why. So this is a strain. So chart here is survival rate using UV radiation and looking at bacillus. So Pumilus here on the far left, we have a different strain of Pumilus. And the strain that we isolated from the clean room, which is Pumilus FAR 0332, that one seems to be extremely resistant to UV compared to the other ones. When we look at UV radiation from a Martian environment, so again, here there was just like UVA, as I recall. Here is more or less broader spectrum of UV radiation that you might expect to see on a Martian surface. Still the same story. I mean, that guy was still extremely resistant compared to the others. I mean, Bacillus subtilis, only after maybe 2 or 3 minutes, it's, you know, doomed. Well, that guy is, I mean, there's like probably only 100-fold decrease, but nevertheless, still much more resistant that other strains. And so the question is, why is that? And so similarly, we did other studies where we were kind of mimicking the Martian environment. And the question is, OK, well, radiation is one element. The CO2 is another element of stress. And the question is, well, when you have those two combined, does that change anything from the survival rate? And so that's more or less here where we kind of expose the organisms to different conditions. We're kind of looking at survival rates. And so here on the plot, we have like desiccations. You have desiccation plus Martian environment for UV side. And what I want to emphasize here is the fact that when you have condition where you have desiccation plus UV condition on Mars, and you compare it to desiccation plus Mars atmosphere plus UV and other, you don't see much changes. And so it seems those two are not additive, which is a good thing, I guess, in some sense. And so that was also very, very interesting. But then we wanted to take it a step further. And what about we expose it to space? And so on the ISS, you have opportunities to expose your samples outside of the station. And therefore, you can expose it to vacuum. You can expose it also having some windows to different environments, like Martian environment in particular. And so we did that. We had some strains which were flown. They stayed in space for quite some time. And so more or less, here is kind of layout of the mission. And so what was interesting, and so you have those coupons where you can put your different strains. And of course, the top layer will be exposed to the environment. Then you have a middle layer and a bottom layer. And so more or less, we were kind of looking at all those different layers. So again, top is in red, middle is in blue, bottom is in green. And then we have all those different environments. And so what you can see is that UV in space is harsher than Martian environment. And then dark is more or less you're protecting yourself from direct sunlight. And you can see that I mean, a lot of those organisms were able to survive. And there was not much differences between the top layer, the middle layer, and bottom layers. And so again, UV seems to be the strongest factor to kind of have an effect on viability. Nevertheless, we still have some survivors that we isolated and certainly studied. And the idea there is to kind of also show that space condition, as I mentioned, seems to be harsher than Martian condition. And again, by being exposed to outside of the International Space Station, that didn't seem to overly impact the overall survival rate of those organisms. So again, super, super resistant. So we kind of try to assess, better understand why those organisms were resistant. And actually, we sequenced the genome. We were expecting to see maybe some specific pathways to kind of DNA repair, et cetera. And actually, many other pathways that you would think would be present actually were absent. And so we still don't have a very good understanding why the organism was so resistant. Nevertheless, so there's been few studies, and some of which kind of related to some of the candidates that might be helping Bacillus pulmonis to be resistant. But what I want to finish with is the fact that this strain is super resistant. But what we did is actually now you have commercial products that are using the Bacillus pulmonis extract as a compound into anti-aging cosmetic compound. I don't have any royalties, so I'm just putting that out there. But there's anti-UV resistant, decreasing reactive oxygen species, increasing DNA repair processes, increasing hydrochloric acids. And so, again, tremendous from going from isolated organisms into the clean room. Now you have a commercially available product. And so anyway, that kind of finished the second pillar. And so now let's transition to space biology. So again, understanding how Earth-based organisms can cope with the space environment. And I'll be taking you to a journey on board the International Space Station. And so, as you're well aware, we take astronauts, we go into space, and maybe at some point, we'll be heading to Mars and so forth. And so, although in some sense, it's kind of closed system, right? And so, in some sense, I kind of bring an environment, which is what we call the built environment. And there have been studies, some of which I've been involved with from the NASA side, where we collected samples from the station at 8 different locations. And as you can see, we kind of find a lot of interesting things. There were a lot of compounds which were more virulent. For example, we isolated Aspergillus fumigatus and showed it was more virulent on zebrafish model. There's a lot of strands which show that actually they were like biosafety level 2. A lot of interesting strands that have actually, when we isolated them and look at the genome, they have a lot of compounds. I mean, as I mentioned, microgravity seems to enhance horizontal gene transfer. And so we saw a lot of genes kind of coming from Salmonella and Staphylococcus and etc. So a lot of interesting work. But anyway, us, among others, have done some studies, and similarly also at the international level, at the European level and Japanese level, kind of try to characterize the International Space Station microbiome. what we did is actually we have an opportunity to fully characterize it, not just from a microbial perspective, but also from a metabolic perspective. And so I'm going to take you onto that realm. So this is the International Space Station. We're able in this time to collect, and we collected almost over 800 different samples from the station, so fully characterizing the ISS. And the idea there was to do two things. One is to kind of sequence into 16S as well as metagenomic sequencing, and then also do mass spec to kind of characterize metabolites, and then kind of generate heat maps of all of those different population, location-wise, and I kind of better understand the environment. I will spare you on the details about the process and the protocol. And so let's kind of focus a little bit on the ISS. So here are all the different modules that we collected the sample. So PMM, which is right here, it's more or less a storage facility. Columbus, it's kind of a laboratory. Node 1, that's where they have a dining table. So more or less the crew can come and have lunch, breakfast, dinner, etc. Well, lab is lab. Airlock, it's more or less where the crew goes before going out. Columbus, I'm sorry. So this is the PMM right here. Yeah, I'm sorry. So node 3, that's where the crew actually exercise. And that's where also the toilet is located. So here it's PMM, which is a storage facility. Columbus is right here. Apologies about that. So this is the lab. Node 2, that's where the crew quarters are. And then this is a Japanese module, and then another storage facility. So we kind of, we were able to kind of collect throughout the station. And the idea there, again, was to kind of fully characterize the environment. And so again, the idea there is to kind of generate a 3D visualization, so at least you can see, hotspots and things of that sort. And so that's more or less what we want to do. And to do so, often, there's different ways to kind of show prevalence or maybe show abundance. And so here, more or less, what we're trying to do is that you select something of interest, divided by a reference genome that we know it's kind of flat and throughout the stations, and then you compare the two, and then you can assess, who's more prevalent and so forth. And then you can use a heat map to generate that. So then the questions, earlier we had like a life support system tool and pool and so forth. So let's dive into that. So the question is, well, this is, the toilet in space. And so the question is, what about we trying to characterize, if there's any fecal matter in the environment, and if so, where, etc. So this is, let me see if that works. Yeah, so you can have a visualization surrounding the, where the space toilet is. And so again, each color codes correspond to abundance. So going from kind of purple to yellow. So yellow is highly abundant, and purple is a little bit less abundant. And again, the toilet is a closed environment. As you can see here, you still have marks on the ruminococcus all over the place. And so, as you can imagine, going to the toilet under microgravity condition is not an easy task to do, right? But certainly our recommendation as a scientist is, okay, guys, you need to do a better job or come up with new ways to kind of mitigate that, right? So that would be, some of the recommendations for sure. And then I guess here we don't see very well, but kind of, show node 3, that's where the toilet is, and that's more or less where you have a largest abundance, which of course makes sense. We can do similar work related to the node 1, which is again where the dining table here. In this case, we looked at lactobacillus. And so again, probiotics, and therefore that could be related to food, which is more or less what we see. So here you have an image of a dining table, and then some things kind of surrounding, which in some sense makes sense. And so again, a way for us to kind of dive into and trying to characterize fully the microbiome environment of the International Space Station. So then we can do some work related to diving into, what's there and all that. So busy chart. So let's focus mainly here. And again here, so you have a phylogenetic tree, and so it's divided by colors, and so these are the phylum. And the color in the inner ring is related to the nodes. And so again, here are the description of all the different nodes. And then the top part is the abundance. And actually, there's 3 different spots right here, one of which is right here, one of which is here, and one of which is there. And those actually are the largest abundance, and those were troublemakers. They were staph, certainly stuff for your interest, Pseudomonas, another one. And so certainly, despite the fact that, they do due diligence to clean, etc., you still have some troublemakers that are out there. I don't think, and so the most interesting part when you do those source tracking bulk of community can be easily related to skin. And so again, very much a lot of things related to human, which makes sense. But also there's a lot of things related to the built environment and material side in particular. The other thing that I wanted to mention is kind of metabolite profile. And so again, we're kind of trying to characterize the metabolite profile. And as you can see here, only 5% of what we collected from the station has a match. 5%. And so, which is interesting in some sense, which means that a lot of things needs to be done in that realm. And here it's more or less kind of looking at detection of different metabolites divided by the different locations, and so the different modules. And so certainly on node 3, it's pretty clear that, which is 3 here in blue, I mean, it's pretty clear you have that kind of unique signature as opposed to the other one. It's more diverse. So you can do similar work related to metabolites. So in this case, like looking like the urine metabolites, so we were able to find phenylalanine, glutamine, only not 3, which is good because that's where more or less the toilet is located. So good job out there. And then similarly, we can look at other compounds. And so again, on the station, there's a lot of build material which is used, one of which is kind of related to rubber, and which makes sense because those are the facility where the crew is exercising and also running. And so you have treadmills and you have like squat machines and so forth. So it makes sense to have those type of materials. So it was kind of cool to see that. And then on the food side, so as you know, crew is congested, so they lose like a lot of sriracha or spicy stuff, and so pepper. We were able to find that all over the place in node 1, which makes sense. Similarly for, actually here's the fungal metabolites also found in node 1 where the dining table is located. And as you can see, I mean, crew tried to do the due diligence to clean the environment and so forth and try to keep it as clean as possible. And so, which was also a good thing to do. So on the metagenomic side, I mean, again, it's a busy side, but I'm not going to dive too much into it. I want to focus to 2 things. One is this one. So more or less here you have different built environment, and we're comparing, for example, houses, hospital setting, COVID, and then the International Space Station. As you can see, the International Space Station and the study that we did, which is 3DMM, is on the far end of the spectrum from diversity comparing to the other environment. So which means that it's pretty unique environment by itself and is an extreme spectrum when we compare it to other built environment. What was interesting, and I know some people are kind of working on antimicrobial resistance. So we did find a lot of antimicrobial in different on the ISS, some of which were actually, and in particular, the beta lactams were kind of high, probably the highest class that we found and come across. And nevertheless, a kind of very broad spectrum, and actually was present in all the different modules. And so certainly it's of concern. We did metagenomics, so actually we were able to fully recover viruses for the first time. And actually, I did inquire if any crew had viral reactivation, and there was crew having viral reactivation. And so those were herpes-based viruses. And it was interesting they were able to find them in the environment. Similarly, we did some similar work, and actually we were also almost able to kind of fully recover the genome of a few usual suspects, right? I mean, you have Kibble-Staph pseudomonas, Pseudomonas, Staphylococcus. I mean, all those are troublemakers that you don't want to be near astronauts. As you know, the crew is immunocompromised. But from an evolution perspective, we kind of collected that guy a few times, and that guy is as well. And we all found it in different settings, in hospital and clinical settings. And here is the numbers of antimicrobial gene counts versus the sources. And as you can see here, all the ISS strains have the highest account numbers. And from an evolution perspective, I mean, we can clearly see that things are evolving over time. And it sounds like those are accumulating antimicrobial resistant genes. And so, of course, it's a problem. So again, this is the largest. So again, we collected 800 samples. And certainly, this is the most comprehensive study looking at genomic as well as metabolomic profile. And I want to point out that we didn't do the Russian side, so that was the other side of the station. And so at some point, it would be interesting to also do that and kind of look and compare. Nevertheless, I think we do have a lot of insights from a built environment and habitat perspective. For example, most likely having a toilet on the far end of the station as opposed to be in the middle might be maybe something to do like that. Things kind of remain in one hand as opposed to kind of transfer throughout different modules. And so anyway, the paper should be coming out in Cell sometime soon. And so. All right, so let's shift. So that's all the people which were involved. So Kate Rubins, for her, I mean, she's the one that was collecting the 800 plus samples on the station. And so it took her a lot of time. And so I just want to thank her. And this is, a team effort with UC San Diego as well as Jet Propulsion Laboratory. All right, let's transition to technology for the next few minutes before we go to diving into the Q&A. And so I've been also involved in that realm. And the reason being is the following. When you do science in space, actually, they're only doing science. They're not doing any analysis. That's a problem. I mean, it takes like 3 years to do an experiment in space, right? And then you do your science on the ground, kind of analyze the data, refine your hypothesis, then you hope to go back. Well, what about if you have capabilities in flight where you do the analysis, get the answer, refine your hypothesis, do another round of studies, and keep going, right? I mean, that would be more efficient, right? But currently, there's limited capabilities on the station. So what about we develop those type of capabilities, right? And so I've been involved in that realm. So the first project I will be diving in is like a few things. One is, if this is of interest to you, I wrote a review paper a few years back now, kind of related specifically to that. And actually, the paper goal was to educate NASA about omics, because at that time, I mean, it was clearly clear based on the conversation that we had, they didn't understand the added value of having those type of capabilities, high throughput methods in space. So the idea of the review was to kind of educate NASA. And so I will be talking about two studies. The first one would be GEM, which stands for Gene Expression Measurement Model. I know some people here are part of the engineering program and working on CubeSats, so that would be diving into that realm. And then the other one is more for having capabilities on the ISS, for example, or maybe lunar surface and so forth, to be able to extract compounds from cells in general. So this is a 1U. So again, 1U means 10 by 10 by 10. So 10 centimeters by 10 centimeters by 10 centimeters. So this is a quarter here, kind of give you the scale. And this system enables you to do a lot of things. One, you will be able to grow cells. Then you have a pump. We'll be collecting the cells. Then here we have a chamber that enables us to do to lyse the cells and then to be able to extract RNA in particular. So we use diamond dust to lyse the cells, and then we use silica-based membrane to be able to collect the nucleic acids. Then, of course, you have like the regions, valve manifold, where you have all the different compounds that you need. So water, ethanol, and etc., to more or less do the processes that you might be doing in your own laboratory. And then we added a microarray. And so the idea there, you extract compound, and then you add some probes combining to the RNA, and then those two hybridize to the array, and then you can integrate. So again, all that is 10 by 10 by 10. So here you have a system, high throughput, enabling you go from growing some cells for quite some time in space, extract nucleic acids, in particular here RNA, and then interrogate how the cells are responding to the environment. Again, that was never flown. So currently, this is just a prototype, which we are TRL kind of 5 probably, so mid-range of a TRL. Now we want to take that to space. So if you guys are interested in collaborating, happy to do so. Then the question, does it work, right? And so before that, when you do extractions here on the ground, there's a few steps that you want to do before you do microarray. And what we did here, we just like remove the amplification part and the labeling part, because that was overly complicated. And so here I'm just showing the system which enable you at one point you can lyse the cells and then be able to collect the nucleic acids. And here in this case you see a green light because we're using cyanobacteria. As you know, cyanobacteria are one of the toughest organisms to kind of lyse, and so we wanted to kind of use that as a model organism. And then we collect our own material. And so in our cases we bypass PCR, so we don't want to amplify. But then what we did, we use a system where we have an array, we have some reporter probes, combine it to the RNA, and all that hybridized to the array, and then we detect through electrochemical detection. And so that's the beauty. So the question, does it work? So here I'm just showing the extraction from the system, looking at the quality of the RNA, it's similar to the ground, and then kind of more or less profiling the expression level profile from the array subsequent to the extraction profile. And as you can see, I mean, it's like 90 plus. tiles a similarity to your ground control. And so again, that could be something to develop and to use. And so I am more amenable to potentially collaboration. Here's another system that we developed, and that system is based on beads. And again, it's automated, and the idea there is similarly is to extract nucleic acids from cells. And in particular, this could be probably amenable for the International Space Station. And so the idea there, we have a magnet, which is collecting. So you have some beads will be attaching to nucleic acids, and you can use magnets, you can wash it and all that. So all are the different steps that is more or less laid out right here. It's something that we tested in the environment. We went to Yellowstone National Park, and then we did some testing. And so what we did here, we kind of show one is how is the device from a quality perspective at extracting nucleic acid. And so we compare that to standard laboratory setting like the Maxwell and etc. And actually the system is better. So here, when we look at different and compare it, so again, Titan is the name of our own system. Maxwell is a commercial off the shelf. When we look at the DNA yield, in almost all the cases, Titan is doing better than any commercially available system, which is, of course, the added beauty. Now the question is, okay, is the system is impacting the diversity of bacteria from a sequencing perspective? So what the next question, what we did, is that we had a mock-up community of different set of organisms, and then we use that mock-up community and ask the question, okay, use the different devices, extract nucleic acid, ask the question, but from a sequence perspective, do you see any differences? And so here we were using different sequencing technologies. So we use Illumina plus the commercial off the shelf, which is Maxwell. MicroTitan is our own system. We just use standards with QIAGEN. And then also we did some nanopores. And again, here are all the different communities, so E. coli, Bacillus, and etc. And if you look at the MicroTitan, which is here in red or in pink, I mean, very close to other system and at sometimes doing much better. And so again, here you have a system which is based on macrofluidics using beads as a mean to extract compound. It sounds like this is working and more actually efficient from a yield perspective, doesn't impact microbial community from a diversity perspective. And so we believe that could be a suitable system that could be used in space to kind of do analytical capabilities and inform the question. And so again, the question is now, okay, now we where you have tools to be able to do science in space, not having to bring back your samples back to Earth and then do analysis. And so the question is, shall we use commercial off the shelf, which is like top notch from an advancement in technology, or shall we use something which is specifically driven towards space? Because as you know, in space, you have microgravity condition, surface tension, diffusion, convection, all those things are different, right? And so in one hand, there's advantage and disadvantage, right? On one hand, you will have like top-notch technology that companies and biotech and all that is developing. But then you don't have all the automation, maybe it's not adapting to the environment. Or that. On the far hand, if you kind of space-driven build, then of course you would be thinking about miniaturization, automation, and all that. But then it would probably take a long time to develop another party. Sweet spot is having those kind of hybrid system, which is kind of combining from both ends to kind of enable those type of technology. And so we believe that we should be focusing on that realm. And so again, the idea there, we think that on a technology roadmap, in some sense, we believe that array-based, sequencing-based, nanopore and so forth, I think that could be used as is. On the proteomic side, we believe that will take some time because it's a little bit more complicated from automations and so forth. And so anyhow, again, here we believe that within the next few years, we can have multi-omics type of capability on the station, but NASA needs to kind of push for full force. It sounds like now they're kind of understanding that and they're realizing that. And so again, there's many different capabilities that can be used. And the idea there is to kind of increase discovery, increase breakthrough. Again, if it takes 3 years to do something that you do and then to go back in another 3 years, well, if you do that in space in real time, change your hypothesis, as I mentioned earlier, then you will get answers and certainly more breakthroughs, and which enable more or less us moving forward from an exploration perspective, but also addressing some aspects related to human health in general. So I will, thank all the people which were involved here, and I think just want to thank you all for being here and greatly appreciate it. And then happy to kind of, have any questions. So thank you again.
generation of mutation. That is by the first, like how is it works, building radiation is born.
Okay. So for people online, there's two questions there. One is more related to the adaptation versus acclimation, and the question was related to kind of better understand what might be the mutation aspects of E. coli in particular, but more importantly, if you combine it to other stress like radiation, how does that impact the overall physiology? And then the other questions are related to radiation as an exposing environment on the International Space Station. So for the first question, I've not done that, but actually it's something that we wanted to do, combine in radiation to symmetric microgravity condition. We did some preliminary work. We didn't see much changes. We've not looked into details about those switches on and off. And again, for us, it was just to kind of compare short-term adaptation to long-term. I didn't show the transcriptomic data. It seems the short-term adaptation is through motility, and I can explain it why, but it sounds like the long-term adaptation was through biofilm formations. And so that's more or less what we saw. But yeah, we didn't look at specifically the switch on and off for those type of mutations. On the radiation side, so yeah, radiation is one of the main limiting factors for exploration, mainly for Mars exploration. Again, it will take probably 3 years, so 6 months to go, you stay there 2 years, 6 months to come back. And most likely you'll be exceeding your current limits from an astronaut's perspective. And so it's either we need to go there faster or come up with ways to mitigate radiation, which is complicated. On the International Space Station, what we did on the crew quarters, so where the crew is sleeping, we added polyethylene panels. So those are compounds with high numbers of hydrogen. So lighter is the compounds, the better it is from a protective perspective, because the problem is that when you have heavy material or heavy compounds, what happens is actually you're creating secondary particles which are more damageful than the primaries, which is a problem. And so radiation is complicated. I think it could be a mix of technology. So on the propulsion side, technology on the material side, and then also maybe some compounds on the pharmaceutical side to kind of help alleviate some of the effects. So I think those three combined might help recruit. But definitely currently estimates is that the crew will be exceeding the current life rate. And so for like 3-year mission is like 1.2 sieverts, and which is far exceeding the current limits on the astronauts. So indeed, it is a big issue, and a lot of work. People are working in that realm. Yes, sir. So yeah, for people online, the question was more related to the ISS built environment and kind of better understanding if those microbes are kind of free living or more or less associated with human activities and so forth. So I mean, for us, what we did is actually we were looking at, so we did some primary work where, of course, when you do sequencing, for example, I mean, if it's a dead organism, where you will still be able to, get material, right? So what we do PMA prior to the sequencing part. So PMA will actually, if the cell is dead, it will block the amplification. In this case, we didn't do that, and so it's possible. But again, we were able to recover almost fully full genome of some of the organism. As I saw, I mean, it's pretty clean environment. And again, from the spectrum, as compared to other built environment, we're on the far end of the spectrum. So I think the crew overall do a good job, due diligence. A lot of the microbes are human related, right? And so certainly things have changed a little bit when there's crew coming in and out. And I think probably there's also some niches here and there because those are surviving, those are thriving. And as I show also, I mean, we were able to detect the same organism all the time. And we clearly see there was an increase in antimicrobial resistance. And so those are actually, so the ISS in some sense also is selective pressure. And so we do see that. But clearly we can see changes when the crew is coming in and out. Yeah.
more beneficial for moon expedition, Mars, where it could take months before we can get the sample, process the sample, and then retest based on device types of things that you guys are building, be very beneficial.
Absolutely, and even more so when you are, as you mentioned, sorry, so the question, I mean, it was not much a question, but it was more a comment, kind of related to the value of having those type of capabilities, because then when you are long duration mission, then certainly you now have the opportunity to send back your samples to do analysis, and therefore having those type of capabilities is critical. So yeah, definitely, and I think this is something which is important to kind of consider. And even more so, as you point out, going back to the moon, going to Mars, you need to have those type of capabilities. And so I think it would be a mix of having those multi-omics type of approach plus AI, machine learning. I think that would be also very, very, very useful. But yeah, we're not there yet, and which is interesting. But then there's also the other component, which is also missing, is the fact that currently the knowledge is related to where we're at, which is the ISS. So 6 months to up to 1 year duration, right? And microgravity condition. Well, you're going to go to the moon. Well, transition to 1.6G, right? And then you can spend more time there, then come back micro G, then come back 1G. You're going to go to Mars. So microgravity for 6 months, 40% of gravity for 2 years, back to microgravity, back to 1G. So, and that transition, so we call that altered gravity. I mean, there's still a lot of questions in that realm. I mean, how things are transitioning, right? And so a lot of work also in that realm, and then trying to kind of mitigate those risks. But definitely we need to have those type of capabilities moving forward, otherwise we're doomed. Sure. Yeah, yeah.
So basically laid it out as you're doing this in space, but the state of the art is not space oriented, so you need to hybridize those two. Is that your strategy? You're going to try to steal some nanopore people or collaborate with nanopore people? Because that seems like the most promising way forward, right? You want that technology, the single molecule sequencing of everything, not just what you've got probes for. You want to be able to combine it into the 10 by 10
Sure.
structure that you want.
Right. So the question was related to what is the added value of using current microarray technology versus embracing sequencing and so forth. So the study actually was founded in 2010-ish. So that's where more or less we started. And that was from the astrobiology program. Then we wanted to transition to space bio and the International Space Station. And actually, they didn't understand what was the added value of having those type of capabilities. That's the reason why I wrote the review, to educate them about multi-omics and what is the added value. And so there's a nanopore on the station. So for the first time, 2016, we sequenced for the first time using nanopore in space. So the first experiment, we're using strains which were isolated on Earth and then took them to space. Currently now what we do is, so from the crew perspective and the health perspective, we monitor the environment on a regular basis. Currently it's plate-based, so we take some swabs, plate it on 2 different type of plates, count the numbers of colonies, and then, oh, this is below the threshold, we're good. Higher than threshold, oh, we need to clean more. But then we kind of send those back to Earth and they do isolates and look at things. Now what they want to do, which is using nanopore, and then take your isolates, try to amplify the material, use it, and do it sequencing. So currently now we're going to that realm, but it's not like automated, takes a lot of crew time, and it is not trivial. Where there, I mean, you can remove the microwave part, add the nanopore part, and here we are, different ball game, right? In some sense. Maybe some questions from online, maybe. Or maybe they can put their mic on if they want to talk. Any question online?
Those hypermutant, I guess maybe you would call them, you found that there were some antibiotic resistant, super antibiotic resistant lines on the ISS that seem to be evolving new lineages, right? So have some of those been brought back, and are they still like super mutants when they get back to Earth, or is this something that it sounded like you had on your radar, that bringing back microbes that have evolved in space could be dangerous as well? I don't know if you guys have explored that yet, or
Yep.
microbes evolved in space coming back.
The question was related to the ISS microbiome and in particular those strains, which seems to increase the antimicrobial resistance genes count all the time, and try to better understand and characterize them. So, I mean, yeah, those were novel organisms. So actually they were isolated from the ISS, and then we fully characterized them. And then actually we found them on Earth as well. And I mean, and you know, I can explain why that's the case, but indeed, it seems like the ISS is adding a selective pressure, and those seems to become more resistant and having a larger accumulation of antimicrobial gene. And so so this is something that, yes, we keep track, and again, we collect samples on a regular basis, bring back to Earth, and trying to isolate them. Of course, we don't isolate and characterize all of them fully because, again, there's too many and etc. But it's something of concern for sure. And so I think now leveraging those multi-omics type of approach, and leveraging now the fact that, you know, this century is a century of biology, and now we have capabilities like using nanopore, I think we are better suited to kind of better understand and kind of have a full understanding of how those are evolving over time. What would be interesting, this is the first most comprehensive study. We did a study in the past where we collected 3 samples at 3 time points, but only from 8 areas. And so we have also a time series of that and how things are evolving. And certainly we saw changes, and changes kind of related to the activity of the International Space Station, which in some sense makes sense. I think for us, it would be important to kind of go back and try to see how things are evolving. But yeah, those are questions which are still question marks about how things are evolving all the time. Despite the fact, again, I mean, this is crew is doing a good job overall, kind of keeping the station clean. And then, you know, there's clean and there's clean. I mean, again, we need the microbiome, right? We need those microbes. And actually, there's more microbes in us than human cells. And so in some sense, we are more microbial organisms than human. But, and therefore, you don't want to be in a situation where you deprive from beneficial microorganisms from a health perspective. And so those are also, you need to find kind of trade-off between, you know, kind of cleaning as much as you can, but still kind of remaining and maintaining a healthy microbiome environment. As you know, I mean, people with asthma, it's mainly due to the fact that early on in their childhood, they were not exposed to different type of microbiome, and because of that, then they become more allergic. And so there's a lot of work in that realm, which is also a concern from a health perspective. Yes? Well, I think going to planets, I mean, going back to the moon would be interesting, but going to Mars, I think that would be very exciting. I think they are those special regions that we believe that maybe there's still some remnant of past life. And I think if the question is, are we alone? No, we're not alone. I mean, again, I think there's what is called the Copernicus principle, which is there's nothing special about us. There's nothing special about the time we live in, and there's nothing special about the location we at. So knowing all those 3 different conditions, you know what happened to Earth. I mean, there's like zillions and zillions of Earth-like planets throughout the universe. And so it is very likely there's someone like us somewhere, or maybe different, but not too different in some sense, or maybe we are, who knows? So I guess, yeah, we'll leave it there. Yes. Yeah, it's true. So it's a colleague of mine, and so a great scientist. And so for people who are interested, he wrote a book called The Next 500 Years. And so the idea there is, what might be a suitable roadmap for us as humanity to become a interplanetary species. And no, I agree. I think those are things that we need to consider. I mean, if we want to, I mean, probably you're aware we're halfway through the life cycle of the sun, right? And so at some point, I mean, the solar system will disappear. And so I think for us, if you want to survive, then we need to become interplanetary species. And so definitely, I mean, certainly from an evolution perspective, I mean, maybe adding some tardigrades genes into you so you can become more resistant, and then transforming and having more like skinny legs because gravity, you don't need your legs, and etc. And so you will look like a flamingo, just like tiny legs, and just so I mean, yeah, I mean, it's possible. It's possible. So yeah, biofilm is a very interesting element within the realm of space microbiology and space biology in general. I think a lot of work needs to be done. I don't know if she's aware, but there have been a few studies, Collins in particular in 2011 and 2013. She looked at Staphylococcus, or maybe Pseudomonas, as I recall, and then trying to look at the overall structure of biofilm formation in microgravity. And actually the structure was very different, and biofilm was kind of forming those canopies, and which was very, very different from Earth-based biofilm formation. So there's a lot of interest there. And I think this is a realm that we still need to kind of work on. And from a health perspective, of course, obviously, as I mentioned, crew is immunocompromised, so they're most susceptible to become sick. And then on the other hand, microbes seems to be more resistant to antibiotics, stress factors, and then having more, biofilm formations due to the lack of gravity. And so I think it is an area of interest, and currently there's a space biology call. And so if she's interested about looking at those aspects in space, then I recommend her to kind of look at it and propose something, and happy to collaborate. All right, thank you. And so again, there's still International Space Station calendars. So you could have, take one if you didn't have any, or if you didn't have any, Begum, good seeing you. Thanks for coming.
Of course.
Get some calendars for you. Yes, yes. Yeah. Yeah. You can take more for the ISS ones for the, yeah, please, please. Yeah. Take. Oh, thank you. Thank you. When did you come? Did you arrive?
Yeah, you were talking about the.
Okay. So like maybe 15, 20 minutes in. Yeah.
What's the plan? Are you still?
Oh.
Yes, my fiance is Spanish.
You have time now? You're gonna lap and then
Uh,
you can drive around. What's your schedule?
students and having pizza. You're welcome to join.
Your time a little bit, another like an hour, then I will go back. We have a step 2 meeting for
What?
omnibus.
Well, it's good that you got invited.
It's not a lot, tissue chips, you will see.
Oh, that one. Okay.
And then 3:30 to 4:30, then I'm all good. I can do whatever you want.
So I will be at the hotel if you want to
I can pick you
me up.
yeah.
Yeah, I am at the Hall 2 Suite.
Here? Yeah, I can pick you up. And then where are you going to dinner? We can spend time until dinner. I can drop you off to the dinner location too.
But I would like maybe to exercise too. So maybe I might need an hour
Or you do
before.
exercise, then I can pick you up because I can stay in
Yeah,
the lab
maybe we
like
can-
by 5:30.
Well, I mean, dinner is at 6, so probably we finish at 7, 7:30. happy to meet after if you want.
Okay.
Or tomorrow I have more time in the afternoon.
Well, we can do some stuff too.
And then
When are you leaving? And how do you
Saturday.
organize? Saturday, okay. Saturday morning, I'm working at the clinic. So I'm happy to just let me know what you need because I'm here. I have a car. I'm happy to drive you somewhere. It's home to sweet.
I will be free probably tonight after 7:30, I think.
Okay.
But it might be late.
No, it's
Or we can have a
where is
coffee.
the dinner place? Downtown?
I don't know. Or we can have a coffee before, like around the. So you say at 4:30 you finish?
The meeting.
can have-
Five,
Yeah. So we
5
can.
o'clock, I can leave the lab.
So we can have a meeting have coffee from 5 to 6.
Yeah, okay, then I can drop you off to your
Well,
place.
they come to pick me up at the hotel, so
Okay. Whatever works. Or then after
so we
you're
can have
done.
coffee. So you can
And then
come
let
pick
me
me
know
up.
where you go to dinner. Yeah, I can. Which home to suites? Where did Fathi go?
Oh, this is.
Sure. So some of that wouldn't have been reimbursed. Right. I think we'll be able to get,
So yeah, definitely we need to.
Yeah.
Hello?
Yeah, I'll send the picture.
Oh, thank you.
Of the email.
All right.
Another paper.
So what time?
It's at 8:30.
8:30.
Yeah.
That's okay. Yeah, okay.
Good. I'm really trying.
How you doing? Oh,
So we were having students same time as to still in this place. So he's a.
yes.
Engineering?
And then we, all the calculators.
Yes.
Yeah, all right, let me know, feedback. Really, if something needs to go. That's good, this is at 8:30.
It's been a while for me.
Yeah.
Oh, you- okay.
I mean, that's when we graduated.
Yeah.
Yeah, thank you.
Okay.
I don't find, have kind of safety, so.
Okay.
Okay, great. Yeah.
That's great.
Tonight,
And I have one more. Oh,
One more?
yeah. I just don't think there's a standard. Thanks for coming.
Sure.
I don't think you're really able to-
I don't think so either. Like, I am stuck in the MBS building over there. USF.
But you're in India.
So you have been here for a while, or did you move?
Yeah, I mean, I.
You're an IB. I know Kerry Scott, I know Jody, but I don't think we ever.
Well, I don't know.
Oh, I see. Okay, that explains
I
it.
work at
Okay.
and I just know which one who's actually running your labs and. Yeah, yeah.
Oh, yeah.
In spaceflight.
Okay.
For patients, so I started. Space stuff.
Oh,
Guys.
okay. Oh, yeah, okay.
Oh, so that's how we
Yes.
ended up inviting.
Yeah, that's good. It's like the movie Martian.
Yes. We'll have it better.
I see.
Sure, yeah.
They give me. Nice to meet you.
Nice to meet you. You're in. Which to meet you. Really? Okay. I don't understand how you like works.
Yeah, you can have.
Yeah.
PGH or the,
TTA, NBC, I mean, we are now capability, but I'm not going to make that now for maintain
Oh, wow. So with the fertility and microgravity? Cool.
from the.
That's a good idea.
Now,
Okay.
Yeah, you very much like being released. It's one of the
Yes, exactly. Yeah, that's.
No,
Space agent stuff.
Okay.
yeah. Okay, so what the students what do you do?
yeah.
But I'm also doing.
Yeah.
And I was like, I'm going north, because I read Japan's name.
Oh, yeah, they've been forming y'all.
Yeah.
Nice.
Let's do some research, actually, yeah. Okay. I'm also looking international, I know you really came there, and I should have these involved.
Yeah, I'm a microbiologist, so. I know, I get emails from them, I don't do microbiome work. I study more fundamental.
I think for me, it's such a,
Need to go to a seminar.
I think it's more.
I need to head out to a seminar. Nice seeing you. After like some weeks.
Yes.
You guys say sometimes, let's say one more live picture. I was looking so high.
Oh.
Like,
Do you want
Like
yeah. Yeah, you know, email, you have to go to find the picture. So you have other plans?
That's just like one quarter of what I brought.
Okay,
I brought like,
well, we'll just keep everything that you use. Well, you probably use it for other.
I'm taking to our laptop.
some more. I mean, those are unfortunately, those are Hispanic. So,
That's cool. You know what? My husband has a bunch of Hispanic students in his class. He teaches in a public school and some of them can't.
so yeah, tomorrow we'll be bringing more. Anyhow, so you might head up with all my goodies on Saturday.
Wait, what? Sorry?
As I said, I brought a lot of stuff. So
Yeah.
tomorrow I'm bringing more for the high school, Saturday for the student,
Okay.
for the teacher. But then anything left, then you can keep it.
Okay,
Just give
Cool.
it away.
got it. Let me see if they're.
Where are you going now, Penny?
Meeting the student somewhere.
Must say
Food
that.
lab pizza. How was the clinic?
Good, now I'm in the lab, so our lab is in the medical building. I'm in the gyno. And then we had the microgravity machine now, the whole. So yeah, we tried to culture, but it was one.
Gravity one you bought, I think.
I had the pulse from Richard Parker. Richard's machine, like the pulse, 3D clinostat, rotating candidate. So we,
Is it the one with Hammond?
I think so, yeah, the inner frame, outer frame.
It's a small one, like that.
Hi. Sorry? Yeah. Yeah.
I think I'm sure.
So yeah,
This one.
first one is Hispanic. I don't know if you. Yeah. So then underneath, you can grab some Steve sure. And then these are all Hispanic. But then you have space station for them. Come to grab.
I have this one.
Okay.
It's somewhere else. Or. What a great arm.
So tomorrow, so when do I meet the students?
Tomorrow in the morning,
Okay.
like 9:15. I think I'll pick you up at 9.
Okay.
Want me to just take it in the car?
Well, but yeah, I would be bringing you told me like 80. I mean, I brought like 100.
There's 80 students,
Yeah, I brought hundreds.
and there's like 10 teachers.
Yeah, I brought it's calendars for them. So And how many teachers?
Probably 10-ish. I
Oh,
guess.
okay. Because you told me-
Oh, no, for tomorrow, tomorrow the teachers will be escorting the students.
Oh.
And then on Saturday, there's another 15 or 20, depending on the registration.
Anyway, yeah, maybe it's better to leave it somewhere and then
Okay. I
we
can
bring
just take,
more.
I'll take it into my office.
Well, I carry because it's heavy. And then I will,
Study too, though.
that's okay, I can.
You don't work out and you're robbing me of mine, so.
I will pick you up at 5 then. Try to find a nice coffee place
Yeah.
close to your hotel.
I need some.
You're gonna see him tonight? Is that you mean?
Yeah, 5 to 6, so you guys
Oh,
will
he's
dinner.
having dinner.
Yeah.
I have no idea, actually, because,
No, but I can drop him off.
That's
That's
it.
what I asked him.
and you could ask, Arash. He's taking him.
You guys can grab everything for them, because I live close to downtown, I can drop him off.
Yeah, I want to just.
Or like we can have a coffee close to downtown area, there are some nice places then.
Okay, yeah, just send an email to Arash.
Oh, perfect.
In charge of the.
Oh, here you go, okay.
So how was the talk?
Amazing, very inspiring, really cool.
All right, so that, and we can go to the office. And so where are the students?
They're in the SCA building. I hope.
Okay, I'm
just gonna wait here. and
Heading
go
back to left, and I will touch base with you.
Yeah.
Nice to meet you. Yeah, nice to meet you too.
You need help with me?
To work on my biceps.
It's like,
So we close the,
oh, I guess we should.
and then close the.
Do you need anything?
Is this your coffee too?
Yeah.
Okay.
So you need to close everything, just leave.
Oh, yeah.